Hi, thank you for having me. It's always exciting to come here and sort of get a front row seat to observe the pace and scope of the research that's being done here at the Allen. So thanks again for the opportunity. Today I want to talk about one of my interests. It's decision making, the neural basis of decision making. My approach to this, the studying decision making, is to train rats using a high-throughput behavioral facility to perform complex tasks. And I use the high-throughput facility to generate very large behavioral data sets that I can use to evaluate quantitative models of behavior and decision making. In addition, I'm interested in the neural circuitry that controls behavior. And so last year, I presented work that I've done developing a new system, or a system for combining high-throughput behavioral training with large-scale in vivo imaging in order to identify the circuits that generate complex learned behavior. Today, I want to touch on one of the themes that has been talked about across the 2 days that we've been here, and that is using atlases and functional brain mapping to tame the complexity of the nervous system. And we're particularly inspired by optical methods for generating high-resolution maps of function across the cortical surface. And we'd like to extend these methods to not just include complex learned behavior and sensory processing, but to include natural behavior, sleep, navigation, social interactions, and also to be able to be integrated with the high-throughput training facility that I just described. And our approach was to generate 2 new technologies that, when used in combination, allow very large field calcium imaging in freely moving animals. The first technology is a head-mounted cortical macroscope. It's basically a demagnifying microscope that can be implanted on the head of the animal. The two key features of this microscope are it's lightweight and implantable and can be fixed to the head of the animal. And the second one is that it offers a very large field of view, 7.8 millimeter field of view at 20 micron resolution. To give you a sense of the scale of this field of view in the context of the rat brain, on the left I've shown the rat brain and the cranial window we use for this surgery. It's an 8 by 8 millimeter cranial window. And on the right is the output of the microscope viewing the cortical surface. The second technology is the generation of strains of genetically modified rats that express the calcium sensor GCaMPf across the cortex and subcortical regions. Here you can see GCaMP expression in the cortex and also the hippocampus, and that expression is bright enough both to produce the maps that we're interested in making, and you can also do it with cellular resolution imaging. So here's an example of combining the 2 technologies together to record a large field of view during, in this case, navigation and also social behavior. The animal here is implanted with our cortical macroscope. This is the first time the animal is wearing the microscope, and as you can see, it's engaging in normal behavior. It doesn't seem to mind the microscope. On the right is the output of the microscope recording from the regions of the visual and parietal cortices. So more recently, we've been using the microscope to record dynamics during an accumulation of evidence task. So in this task, the rat is trained to count visual flashes and to identify the location of a reward based on differences in the number of flashes. So the animal is performing an accumulation task. Here I'm showing in the bottom portion of the field of view is the visual cortex, and in the upper region of the field of view is the parietal cortex. Now there's a couple interesting things to point out in this movie. The first one is that we see complex spatial temporal dynamics throughout a trial. This is the average across 150 trials. And the second one is that in contrast to some of the experiments that we heard about earlier today, imaging in head-fixed mice, we see a dramatic reduction and sculpting of the activity throughout the course of the trial. Now, I'm not sure at this point whether that's due to the fact that these animals are unrestrained or potentially the fact that the task demands are different, but it's an interesting point of comparison. And if anyone would like to talk more about the dynamics that we observe, or if anyone's interested in using these technologies, please talk to me afterwards. And with that, I'd like to thank my collaborators. Stefan Thieberge and David Tank developed the first generation version of the optical path that we use for this microscope. And Alla Karpova and colleagues at Genelia Farm were collaborators in the generation of the transgenic rats. Thank you.